Hello, my students. This is another section of the lecture slides for Chapter 9. In this particular lecture, the focus will be on the process of meiosis in terms of how it can generate genetic variation and also cause mutations to occur, which can result in genetic disorders. Now, before we begin, one of the things that I would like you to do is to download the lecture note sheet, which is based on slide number 2, that looks like this. All right. What I would like you to do is to print it out and then work this out at your own pace so that you can try to figure out what is the normal process of meiotic cell division, sketch and or draw any one of these chromosomes out, and then try to figure out what happens in each of these two latter cases where non-disjunction will occur, meaning where a mutation occur, when the chromosomes do not divide equally between each of the respective daughter cells. So once you have done this, then you can continue and watch the rest of the lecture. I do believe that this would be much more meaningful to you. And if we were in class together, I would have set aside about 10 minutes for you guys to try to figure this out. I would recommend that you use a pencil to draw this so that if you make any mistake, you can just easily erase it without the trouble of having to look for whiteout or to reprint yourself another copy. All right, so let's begin. Assuming that you have already completed your assignment, then let's talk about how this actually happens. So in the process of meiosis and sexual reproduction, one of the beauty of it is that there are several mechanisms involved in making sure that the future daughter cells would always be different to a various degree compared to the parent cells. For one, all of the pairs of homologous chromosomes that you inherit, one a set, or I should say half a set from your parents and the other set from the other parent, when they come together, you really do not know how they're going to be combining. And for example, each of the 4 sperm cells that the father produces, each one of them can contain variations of different combinations of his genotype. And by the time you receive the 1 egg cell from the mother, you also do not know which one of the 4 daughter cells become the ovum, such that when the sperm and the egg cell fertilizes together, you have a unique set of DNA. During the process of making these sex cells, in the process of prophase 1, something exciting happens. For example, crossing over, where in the previous lecture you have seen how portions of the genetic material that controls the same body traits from the mother side and from the father side, when they line up in pair during prophase 1 and they form a tetrad, they can actually swap some of the genetic materials that control the same body trait, which results in variations or similarities in body traits from both the mom and the dad when fertilization does occur. Then there is also the fusion of gametes, as I said a minute ago, where you don't know which sex cell is going to be selected or comes together to complete the fertilization process, such that an embryo would be formed. Now, in this particular case, let's take a look. So this is just a very quick review. What you're looking at is a set of a body cell right here that has been replicated prior to meiosis. During the process of meiosis, the pairs of homologous chromosomes would then line up. Now, again, we have 2 different colors that are used to designate one set from the mother and one set from the father. The same is true on this side. Now, even if you do not think about crossing over immediately, the alignment or how this is going to be paired up during the process of prophase and metaphase, you can see right away that right here on this side, you have 2 blue chromosomes lining up on one side of the cell and 2 red ones on the opposite side. But then in another daughter cell, you have the switching, the alternating of colors, where you have a blue and a red on one and the red and the blue on the other one. Just the fact that you have them lined up differently in a different order, and if you trace the results of the subsequent daughter cells, you can see that you would wound up with different options in terms of what kind of genetic material would be passed on in each one of these respective sex cells that you see here. Now, in the first 2 over here, you would have a quarter of a chance of resulting in dominant alleles, all in each one of these 2 sex cells, or all fully recessive alleles in these latter 2. However, in the other scenario where you have the change in alignment between the two different color chromosomes, you would wound up having the expression or the inheritance of different combination of alleles. This gamete, if it were to be inherited, you will have a dominant allele and a recessive allele, and the same is true for a second one. But then you also have an option or alternative where you can have a recessive R allele and a dominant Y allele, and the same is true for the last one. This is what we call principle of independent assortment, which I will go over in greater detail when we begin to talk about Mendelian genetics in chapter 10. The idea is that for each pair of genes that control a different body trait, for example, the capital letter R's and lowercase r's control the coloration of the seed shape in a plant, in a pea plant, and then the letter Y's designate the seed colors of a pea plant. They could all be reshuffled without the influence of the other one. So you can form new combinations of colors and shape in the resulting plant. Now, during the process of crossing over, you can see that genes actually get swapped in the same locale, because that's actually where they are controlling the same body traits. So in this particular case, what you're seeing right here, this is the centromere, right here in this big oval circle. This particular chromosome, it's orange in color. This one is yellow coming down, but then various sections of the area where these genes actually touch in this particular case, in the lower portion of this particular area, this is where they would swap the genes, therefore resulting in different color combinations or different mixtures of genes that could be passed on to the next generation. Now, this particular image, I do not expect you to have it memorized. The whole idea, the reason why I wanted to even show you this is because for many of you who are advanced learners and really curious about how geneticists actually communicate or figure things out, remember we talked about P arms and Q arms in a chromosome. It's a chromosome structure. We label different portions of the arm or the chromosome in this case. It's so that it makes it easier to facilitate the discussion process and also the research process to figuring out where do each segments of the gene and where are they located even on a chromosome. So these are actually kind of like the addresses, if you will, what we call a physical map of various genes and their distributions along a chromosome. And depending upon how far away they are from the centromere, geneticists can actually use it to calculate what are the odds in which crossing over will occur between some of those genes. And the closer they are to where the centrioles are, sometimes it's less likely for them to carry out crossing over. Now, this is a summary slide that I had shared with you during the Chapter 9 Lecture C video. And this is just to give you a quick reminder in case you're finding this to be a bit overwhelming and that you just want to have a reference chart. But let's go on, assuming that you understand all this, let's go ahead and take a look at the rest. So what happens when things abnormal happens? Say, for instance, you're exposed to excessive UV radiation, X-rays, and various other forms of environmental pollutants. It can actually lead to an uneven distribution of chromosomes in a process known as nondisjunction. And so how does it occur? So the question is, what happens if the spindle fibers distributed or separated the chromosomes unequally as you have learned it in normal meiosis 1 and meiosis 2? Nondisjunction is defined as chromosomes that fail to separate properly during either anaphase 1 or anaphase 2. If you recall, anaphase is when the cell elongates and pulls the chromosomes apart. They could be in homologous pairs, like in the case of anaphase 1, or they could be just double-stranded chromosomes that are aligned in single file during anaphase 2. But sometimes what can happen is you could have spindle fibers that pull an extra full chromosome to the opposite side of the daughter cell, resulting in errors and mutations in the subsequent daughter cells that are produced. Down syndrome is one of these examples. Down syndrome, also known as trisomy 21, the name is derived from the fact that a person that is born with this condition has 3 copies of chromosome number 21. Therefore, trisomy, tri meaning 3, somy referring to the fact that these are somatic cells that is going to be affecting. So these are regular body cells, not your sex cells. So what are some of the forms in which chromosomes may become damaged? Well, it could be caused by a deletion of a section of the chromosome. So you can have portions of the genes that are missing, that are pulled off. You could also have something that is called translocation, in which a section of the chromosome, it's reattached to a completely different chromosome that it's structured to manage other body traits. But then you could also have something called inversion, where the sequence of genes, such as A, B, C, D, which is the proper order of the alphabet as we know it, they may get reversed, such that they would be put in the order of CBA instead of ABC. And if you were an enzyme, you're trying to read this genetic information, the information would not be accurate, therefore producing a protein molecule that could potentially be harmful to your body. Or in some cases, if you're lucky, it may not result in any real serious change, or not something evident at least. But we're going to come back and explore all 3 different types of major chromosomal mutations and gene mutations later on when we get into molecular genetics. So I gave you this particular worksheet. I hope that you had a chance to work it out. Let me show you what the answers are. If you take a look at this particular diagram, what you will notice is that under normal meiotic cell division, there are here at the beginning 4 chromosomes, 1, 2, 3, 4. And they form 2 pairs of homologous chromosomes. They are lined up based on the structure and function that they manage. And then during meiosis, at the end of meiosis 1, each one of these pairs will then be separated from each other, resulting in 2 daughter cells, each one having a double-stranded chromosome, each one. So at the end of meiosis 1, which we call the reduction division, your diploid number of cells becomes haploid at this point. But because each one of these are still double-stranded chromosomes, we need to move it along and then further divide it one more time, such that each of these double-stranded chromosomes now becomes single-stranded. And if everything works out properly, you will see that the 2 double-stranded chromosomes become single-stranded, resulting in each one of these daughter cells having equal number of single-stranded chromosomes. So I have 2, 2, 2, and 2, but because we started off as 2n equals 4, down here we have n equals 2 chromosomes. Now let's take a look at what happens during non-disjunction in meiosis 1. So during this particular process, instead of having 2 double-stranded chromosomes, each moving towards one end of the cell or one side of the cell, down here, 2 of these double-stranded chromosomes got shifted or pulled to the opposite side, resulting in this side of the cell having a missing chromosome. So the rest of this daughter cell will then carry on meiosis 2 normally. Then what happens is this side of the cell that pulled an extra double-stranded chromosome here will then divide it properly, but then all of the daughter cells would have 1 extra chromosome. And this is how the trisomy condition can occur, where you have 3 copies of the same gene coming up, all right, or the same chromosome. Now, this particular side of the cell where you only have 1 chromosome left will then also be carried on normally as usual, and then divide it in half. But notice the formula for this is n minus 1, because the normal haploid number is missing 1 chromosome because it was pulled by the other one. Now let's take a look at what happens when non-disjunction occurs in meiosis 2 instead, meaning the mistake happens in the second half of meiosis. So under meiosis 1, everything is divided normally, and if you move over to the normal side, the picture is actually identical. But then what happens here is that you can have a full chromosome that is not fully divided properly. So again, resulting in this side of the cell having 3 chromosomes, and then the opposite side is missing 1. Luckily, you still are able to have a daughter cell that has resulting normal cells, normal chromosomes, such that it would result in a normal haploid number. Now, in this particular case, when meiosis, when the non-disjunction occurs in meiosis 2, you can actually still have 2 normal gametes, right? Look at this, N, they're both normal, n minus 1 is not normal, n plus 1 is not normal. But if meiosis 1 is the area where non-disjunction occurs, look at the resulting gametes. You have n plus 1, not normal, n plus 1 not normal, n minus 1 not normal, n minus 1 also not normal. You cannot actually have any normal gametes if this particular person ends up having non-disjunction meiosis 1. So a typical question that can occur on an assessment could be that which one of these two types of non-disjunction could be more detrimental to the future offspring? And the answer is obvious. It would be non-disjunction that occurs early on in meiosis 1. So what are some of the genetic diseases or disorders that are caused by non-disjunction? Let's look at some examples now. If you look at chromosome 21, here are some of the bodily characteristics and diseases that are associated with the genes that are found on chromosome number 21. Now, obviously, you will not be expected to memorize or know any of this. This is just for your references only, and I thought that it would be interesting for us to kind of take a look how far research has been able to do for us to gain a better understanding of how genetics can control the formation of the human body. So let's take a look at Down syndrome. Now, Down syndrome has a number of really obvious features and characteristics. Once you learn to identify what some of these facial features and bodily characteristics are, when you walk down the street and you take a good look at people that pass by you, you may actually become very good at identifying people who are born with this particular condition. In the United States, there are about 6,000 babies that are born with this disease annually. Now, when you take a look at this particular image, obviously this is a drawing, but there are a few features that should stand out to you almost immediately. For example, if you look at the face of this baby, the eyebrows are further apart. This area between the eyes look like it's much further away from each other than what you would consider an average looking face would look like. That's in part because this particular baby typically have a flat nose, so that it allows you to have this misconception that they have a wider spans between the two eyes. The eyes also have a tendency to have this upward slant right here and also right there. They also tend to have smaller ears, a small mouth, and there are some characteristics about the hands that are interesting too. They tend to be wide, but shorter hands. And in addition, in the hand, there is actually a single crease that cuts across the entire palm of your hand, all right? And then the toe, the main toe on the foot is actually much further apart compared to the other digits that you would see over there. Now, let's look at a real image. This is what a Down syndrome, a baby with Down syndrome could look like. And again, those features that we talked about before, the distance of the eyebrow between the eyes, the flattened nose, these are very telltale signs. This particular graph is really striking. And it is also something important if you ever decide to start your own family in the future. If you look at this graph, for somebody who is around 20 years old, and you move your chart upwards to about 34 or 39, you will notice that there is a pretty significant change. And well beyond 39, the chances for a woman to develop Down syndrome, I should rephrase, for their baby to develop Down syndrome is much, much higher, in part because mutations begin to accumulate and occur inside the woman's body. Don't forget, a woman or a female is born with all of the egg cells that she could have at the time of birth. So the later she chooses to have her baby, the older the egg cells would be that would have been stored in her body. So what are some of the mechanisms or ways in which we can try to figure some of these things out, such that if a woman does wind up having a child later in life, they can actually prevent maybe giving birth to a child with Down syndrome, or that they can decide whether what should they do in taking care of a baby with this particular condition. In modern society, we have a new prenatal screening technology called cell-free DNA, also written down in shorthand cfDNA. And what happens here is that, as you see in this diagram, as the embryo is developing inside the mother's body, once it gets past to a certain number of weeks, and in this case at about 10 weeks of pregnancy, some of the baby cells would then go through the normal growth process, just like a normal human child would after they are born. Some of the cells would slough off. From some of the cells that slough off and out of the baby's body, it would be suspended inside the amniotic sac. And then what happens is portions of the cells will break apart, releasing the genetic material that was contained inside, and it actually would make its way through the umbilical cord into the mother's body. So what the doctor typically would do is they would get a sample of the blood from the pregnant woman. And then from there, you can distinguish which portion of it is the mother's DNA and which portion belongs to the baby. And of course, one of the interesting things that doctors can tell right away is whether the child would be a male or female based on the sex chromosomes that they can see. But what's also interesting is they can also do a count, meaning counting the number of chromosomes that are found in the cells of the baby. So this particular technique is typically used to detect things like Down syndrome, trisomy 13, trisomy 18, and various other conditions. You can also use it to find out, obviously, the blood type of the child. Now, this particular procedure, it's highly reliable, with a very low percentage of false negatives that it would generate. However, if during the screening process, the doctor finds out, there's a chance, I think, that the baby has one of these major genetic conditions, then they would then invite the pregnant woman to return to the office again and perform another set of screening process that has been used for a very long time. Some of you may know these particular techniques. So before I proceed with the next one, what I would like to do first is to show you the video about prenatal screening of cfDNA. Now, I have provided the link of this for you in the description section of the YouTube channel. Hopefully, if the sound quality on here is not so good, then you can use the ones from YouTube directly to view this.
Prenatal cell-free DNA screening, also known as cfDNA screening, is an optional prenatal blood test that screens for certain genetic conditions. It can be performed as early as 10 weeks of pregnancy. cfDNA screening goes by many different names, including non-invasive prenatal testing, NIPT, or non-invasive prenatal screening, NIPS. You may have heard it referred to as the new gender test, or be familiar with specific test brand names. Our blood contains fragments of our DNA, known as cell-free DNA. When a woman is pregnant, her blood also contains DNA fragments from the placenta that has a genetic makeup that is usually identical to that of the developing baby. By analyzing this DNA in mom's blood, it can be determined if there is an increased or decreased chance for certain genetic conditions in the baby. Though it began as a screening for Down syndrome, over time, the list of conditions that can be screened for has grown. Not all cfDNA companies screen for the same conditions. These conditions are all very different from one another. It is not always possible to know before a child is born how much their health will be affected. It is important to know that cfDNA screening cannot detect all genetic conditions or birth defects. No test can guarantee the birth of a healthy baby. Prenatal cfDNA screening is a screening test that can determine if the chance of certain genetic conditions in a pregnancy is higher or lower. Since cfDNA screening is not a diagnostic test, it cannot give you yes or no answers. If a result indicates a high or increased chance, then a diagnostic test, such as amniocentesis, can be performed if desired to determine whether the baby really has the condition or not. If the results indicate a low or decreased chance, the possibility the baby has the genetic condition is lower, but not zero. cfDNA screening has been shown to be more predictive for Down syndrome compared to other screening tests, such as maternal serum screening. However, similar to maternal serum screening, it is possible to have a cfDNA result indicating a higher chance of a genetic condition, even though the condition is not present in the baby. cfDNA screening is performed on a sample of a pregnant woman's blood. While there is no risk for pregnancy complications, such as miscarriage, it is important to think about what these test results may mean for you. Most of the time, the test will come back with a low chance for a genetic condition, and many women feel relief based on these results. However, if the test indicates a higher chance of a genetic condition, it may create worry and uncertainty about what to do next. cfDNA screening has some benefits and drawbacks when compared to other screening tests, such as maternal serum screening. Decisions about genetic testing in your pregnancy should be based on your own beliefs, values, needs, and personality. See our video, How to Decide About Prenatal Genetic Testing, as you consider your prenatal testing options.
So let's take a look at what some of those techniques are. One of these techniques that you could tell, it's called the transcervical chorionic villus sampling. In short, we sometimes call it CVS. That begins with the letter C from chorionic, V from villus, and S from sampling. The chorionic villus sampling is produced where you would use where you use an ultrasound to monitor the positioning of the embryo, and then what the doctor would do is that they would put a catheter, a soft plastic tube, through the woman's birth canal and into the area known as the chorion. Now, this particular area of the tissue is actually created from the baby's DNA. So when you take a small sample of it, you can then use it to perform a karyotype, and you can count the total number of chromosomes that the baby actually has. And if there is some form of trisomy, you can easily identify it using this particular technique. Now, this procedure is a bit invasive, and there is a 0.1 to 0.5% chance, or 1 out of 100 or 200 babies when this technique is performed, that can lead to a miscarriage. This technique is actually performed a little bit later when the baby is approximately 10 to 12 weeks old. Another technique known as amniocentesis could be performed a little bit later beyond this time period, and it is done between 15 to 18 weeks. In this particular method, what the doctor would do is, again, they would use ultrasound to monitor the positioning of the growing fetus. We now call it a fetus because any embryo that is beyond 8 weeks, that's what we call it. And what you do is you take a syringe, you would try to use it to capture some of the amniotic fluid such that you can hopefully pick up some of the fetal cells that has been sloughed off from the baby's body, and then again perform a karyotype to count the number of chromosomes that you can find. And hopefully, if there are some form of mutation, you can see it quickly and allow this information to be passed on to the parents, and then they can make a decision or make preparations for caring for a child that has a particular condition. This particular procedure, it's also an invasive procedure that carries a higher risk. What it does is that there's a 0.3 to 0.5% where it can lead to a miscarriage. And there could be even higher risk if you try to do this before 15 weeks. So what are some of the other conditions that non-disjunction can also lead to? You could also have a condition known as the Prader-Willi syndrome. The Prader-Willi syndrome, it's a little bit different. Now, you take a look, it's actually much more evident if you look at the image of the baby here. Take a moment. Can you see what's a little bit different about the image, the photograph of this baby, compared to most of the normal baby that you may have experienced encountering in your real life? So one thing you may notice is that the baby seems to be overweight. If you thought about that, you are correct. You may also notice that the arms of the baby looks a bit more segmented, in part because of the excess fat fats that it has inside of it. And the fist that the baby that is shown in the image also tends to be disproportionately smaller compared to the rest of the baby's body. Prader-Willi syndrome actually has a number of conditions. It can be caused by it can cause floppy skin and tail. They could be slightly mentally challenged. They tend to be much larger in body size because of the excess fat content that they have. They tend to have tiny hands and feet. They tend to be shorter in terms of body height and underdeveloped sexual organs. And they also tend to have particularly strong temper tantrums if you refuse to give them food. Now, there is another condition called Angelman syndrome. In this particular case, it's a bit different. Both of these conditions, Prader-Willi and Angelman syndrome, have a problem with a mutation on chromosome number 15. But the difference is that the Prader-Willi syndrome the mistake is caused by a missing paternal piece of chromosome, whereas in Angelman syndrome, it's caused by a missing maternal piece of the syndrome. Now, the difference between them is caused by something known as genomic imprinting. I have a video that I want to show you next to explain this in greater detail. But to go back and contrast these two, in Angelman syndrome, individuals who inherit this particular genetic disorder tends to have spontaneous laughter. They tend to have jerky body movements. They are much more mentally challenged, and they also have other motor and mental symptoms that may be more recognizable. So what are some of the body traits that chromosome number 15 can control? Well, here is a long list of some of the examples of other diseases and problems that is associated with chromosome number 15. And if you take a look at this, this is a normal pair of chromosome number 15, where P designates a chromosome that it was inherited from paternal, from the father, and M stands for maternal, meaning that you inherited this chromosome from the mother. In this particular case, these two chromosomes are normal. But sometimes what can happen is you could have a portion of the paternal deletion happening right in the Q arm. That's on chromosome number 15. That's where this Q's come from. It means this section of the DNA of the chromosome itself, and it could be missing. Now, in about 65 to 75% of the cases, this is where the error occurs. But sometimes you could also have imprinting defects that would be responsible for these 2 to 5% that you see here. So how does imprinting work? And how does it lead to these genes from not functioning properly such that it results in different forms of syndrome? Let's take a look.
Hey everyone, today we'll be taking a look at quite an interesting topic known as genomic imprinting. However, before we dive into that, let's start off with a little bit of background information as to what DNA is and how it works within organisms. Now, DNA is also known as deoxyribonucleic acid. It is the blueprint for the human body, and it tells your body how to grow and make sure everything is in the right place. It can determine whether you are a boy or a girl, or if you have brown hair or black hair. DNA contains genes, which carry the information for a specific trait. A trait is a feature or characteristic that makes up who you are. For example, hair color is a trait controlled by a gene. The genes that control all of your traits are passed on to you from your parents. Now, DNA is normally very, very small and is found in the cells of your body. Now, it is often packed in structures called chromosomes. These chromosomes look like an X. Humans have 46 of these chromosomes, and they usually buddy up into 23 pairs of each, for a total of 46 chromosomes. inherit one set of chromosomes from your mother and one set of chromosomes from your father. Genes control traits is by expressing themselves to the cell and then telling what proteins to make and how much of them to make. So, genes are the blueprints or the instructions, and the cells are the factories. But the interesting thing about genes is that they're also a lot like a fan or a light bulb. When you don't need them, they can be switched off. And when you do need them, they can be switched on again. For example, after you are done growing, the genes that control food will be turned off because you don't need them to grow anymore. Now, there are many mechanisms through which genes can be turned on and off. In some cases, the switches are temporary. But in many cases, genes can be turned off permanently and will never be expressed. Okay, so what is genomic imprinting? Sometimes we're born with genes that are already turned off depending on which parents they come from. This phenomenon is known as genomic imprinting. But remember, we receive 2 copies of every gene, one from our mother and one from our father. Genomic imprinting is simply where a specific gene copy from one parent is turned off, while the gene copy from the other parent is turned on instead of both being on together. For example, if your father's genes are imprinted, then your mother's genes are turned on while your father's genes are turned off. This would mean that only your mother's genes are directly influencing how your DNA is being expressed. Those genes are called maternally expressed genes, or MEGs. Alternatively, if the mother's genes are imprinted, then only the father's genes are expressed and are called paternally expressed genes, or PEGs. Now, imprinting occurs due to the modification of DNA. The DNA is modified so that specific regions of it are unreadable and thus are not expressed. It is important to understand that imprinting does not change the sequence of DNA, but merely changes how it is being expressed. Now, imprinting is necessary for the normal development of many some instances where imprinting causes issues. Some imprinted genes encode very important proteins or have important roles in regulation. If something goes wrong with their remaining active copy, certain diseases may occur. Look at that turtle. Doesn't he look delicious? Imprinting disorders can occur in many different ways, but the most common way occurs when imprinted genes are deleted from their chromosome. Inside the cell, there are many processes that happen all at once. When the cell is making DNA, mistakes can happen. One type of DNA mistake is called a gene deletion, where a portion of the chromosome gets cut out. When parts of genes get cut out, those genes become inactive and are essentially turned off. For genes that are not imprinted, each copy from the mother and father is equally active. So, if one of them has a deletion, the other copy is still working and can make up for the loss. However, if a gene is maternally imprinted, which means that only the father's copy is the one that is usually turned on, then a deletion in the father's copy can be very dangerous because the mother's copy is already inactive and cannot help. So in this situation, both copies of the gene are off, and that whole gene's function is lost. This can cause disease. There are many different diseases and conditions that involve imprinting disorders. Examples include several types of cancers, diabetes, obesity, schizophrenia, bipolar disorder, autism, Angelman syndrome, and Prader-Willi syndrome. Those last two syndromes are prime examples because they are both related conditions that are primarily influenced by imprinting malfunction. All right, so Angelman syndrome is a neurodevelopmental disorder characterized by sleep disturbances, seizures, jerky movements, and frequent laughter or smiling, while Prader-Willi syndrome is characterized by low muscle tone, reduced physical development, cognitive disabilities, and obesity. Studies of Prader-Willi syndrome in particular have revealed a lot about genetic imprinting and the functions of genes that are imprinted. One study found that paternally expressed genes on one chromosome normally regulate the activity of paternally expressed genes on another chromosome. In Prader-Willi syndrome, deletions in the father's genes cause deregulation of the mother's genes, and when those maternal genes are expressed without regulation, their gene products may be the main causes behind the symptoms we associate with the syndrome. So not only are imprinted genes important independently, but they affect each other and interact in order to maintain proper cellular and bodily functioning. There is still a lot left to be understood and discovered when it comes to genetic imprinting and gene interactions. While this video does not cover every aspect of imprinting, we hope it has provided an interesting introduction to a complex and vital phenomenon.
So I hope this particular video was helpful in helping you understand what is genomic printing and how it could lead to genetic diseases. Here's another example of what can happen during non-disjunction. In this particular case, what I'm talking about is called Klinefelter syndrome. So this is a man who has inherited an extra X chromosome. So we call this XXY. And in this particular case, what ends up happening is with the extra X chromosome, this particular person will wind up being a bit taller than the average male. He would have less facial hair, also less body hair. There are signs of breast development. There is cases in which they can develop severe osteoporosis at a much earlier age. There is also feminine distribution of fat and also a smaller size testes. Now, on the other hand, another different condition called Turner syndrome is where a female is born with only one X chromosome. So this person has a total number of 45 chromosomes as opposed to Klinefelter with a total number of 47. Because of the absence of a Y chromosome, this person is still considered to be a female. And some of the traits and characteristics of Turner syndrome includes a shorter stature, a lower hairline in the back, and I have an image of this for you to see later. There is also an extra fold in the skin near the neck region. Some of the things that are not as obvious includes there's more tend to have a larger constriction of the aorta from the heart. The elbows tend to be deformed, and the arms tend to be turning more in outward position. In addition, the nipples on the female body tends to be more widely spaced. The ovaries is also underdeveloped, such that reproduction becomes a problem as the female gets older. And she tends to have smaller fingernails, a shortened metacarpal, and brown spots all over the body, and also no menstruation. So if you take a look at this on this young child, you can see quite evidently that around the neck region, there's a lot more skin that is more widely distributed right here and right here. Whereas if you were to take a look at yourself in the mirror, you will notice that much of your skin ends right around this particular contour that hovers around where the muscle masses will end. And when you take a look at the back of this young lady, you can see that the hairline is much lower. And if you look at yourself or just put your hand behind your neck, you can feel that your hairline tends to be higher in comparison. Um, this particular webbed neck is a condition that's associated with Turner syndrome. So here, this is the end of the lecture for non-disjunction. I will be providing with you some form of review sheet that would help you review some of this material, and hopefully you can also generate some questions that you can ask me during our open sessions, which I will schedule and announce in advance. If you cannot join me at those open sessions, don't worry. You're always welcome to email me at your convenience at a time that it's much more comfortable or convenient for you. I look forward to seeing you guys sometime very soon. And if not, hopefully I can hear from you online. All right. Take care, everyone, and be safe.